Welcome to the latest installment of the AST Journal Club series. Today's session features the Journal of Clinical Investigation article, Host-versus-commensal immune responses, participate in the rejection of colonized solid organ transplants. This session is hosted by the AST's community of transplant scientists. In a moment, I will turn our session over to our moderator, Dr. Andrew Wells from the University of Pennsylvania. However, before we begin the main presentation, we have a few housekeeping notes to help you engage with today's session. There is currently a viewership polling question displayed for the audience. Please take a moment to answer this question while we finish the remaining announcements. This journal club is being recorded and the archive will be available on the MyAST website 2 to 3 business days after the live session. Please note that all of your lines have been muted so that only the presenters can be heard clearly for the archived recording. If you have a question for our panelists during the journal club, we encourage you to participate by using the Q&A button in the Zoom webinar panel. Questions submitted via the chat window may be missed during the presentation. If there are questions we do not have time for, we will either answer them individually offline, or we will post the full question with the answer on the website following the journal club. Finally, when you log off at the conclusion of today's journal club, you will see a link to a short evaluation survey to complete. Please fill out the survey to help us keep our content current and engaging. I will now turn the session over to our moderator, Dr. Wells, to begin the presentation.
Thanks, Brian. I'm Andrew Wells. I am representing the community of transplant scientists, and we today have asked Isabella Pirozzolo from the Columbia Vagelos College of Physicians and Surgeons to present her exciting work that was published last year when she was in the laboratory of Dr. Marisa Alegre at the University of Chicago. Again, as Brian said, please enter your questions into the Q&A panel. And during the Q&A session, I will draw from that panel and moderate those questions at the end. Without further ado, Isabella, I'll send it over to you for the presentation.
Great. I'm just going to X out of this poll. Give me 1 second. All right. Well, hello, everyone. I'm really happy to be talking to all of you today about our article that came out a little while ago in JCI. So before we start, so I have no financial relationships to disclose, neither does Dr. Alegre. Dr. Wells has shares in Johnson & Johnson. All right, so I'm talking to a room of people who know a lot about transplantation, probably more than I do, but I'm going to start here. So when we think about transplantation, we think of it as, this life-saving measure for people with end organ failure. And the problem, as we all know with transplantation, or one of the problems, is that on these donor organs, which typically, most always, are allogeneic grafts, meaning that they are genetically distinct from the recipient, these organs come into the host, and the host immune system does exactly what it's supposed to do, which is attack things that aren't itself. And it mounts an alloimmune response with alloreactive cells that attack the graft and ultimately kill it. And this is why patients take immunosuppressive drugs to dampen this immune response. And this is sort of the conventional wisdom about how we both what goes kind of wrong, or what immune response drives transplantation forward, and also why we need to combat it. But what we know is that it's really not that simple. Like, you would think if that was in a vacuum and in aggregate across the population, you said, okay, let's assume that across all organs, everyone's genetic differences are the same. You would expect that if that's all that's really going on, that outcomes would be pretty similar, but we know that's absolutely not true. So what you can see is that there's really striking differences within 5 years after transplantation of how different organs do by organ type. So for example, the lungs and the intestines, about 50% of them will be gone by 5 years after surgery. So their half-lives are much shorter than something like a heart or a kidney. And you can imagine a lot of reasons why this might be. These grafts are different sizes, so maybe there's a different antigen load. They're made of different cells and cell types. So some of those cells might be more immunogenic. Barrier organs like lungs and intestines come over with their own subset of immune cells. from the donor. And so you could imagine that augmenting the immune response in some way. Of course, these organs are exposed to more just infections that we know can break tolerance, toxins, the surgeries are different. You can think of a lot of different differentiating factors. But one thing that's very, very clear is that if you look at these 5-year survival rates, they correlate exactly with the degree of colonization of these organs. So with intestines being the most and the heart's the least colonized with just commensal organisms. So things that are on you and me every single day, bacteria and you know, usually we're just in a steady state with. And so we took this observation and we asked, first of all, are colonized grafts rejected faster than sterile ones? And so the way that we set this up was we said, okay, we're going to take the same organ type, we're going to you know, make it the same size graft and all of these mice with the same environment. And we're just going to do one thing. We're going to paint the donor skin with the common human skin commensal Staphylococcus epidermidis. And we're going to restrict this colonization to just the skin using the antibiotic vancomycin, which is going to kill any bacteria that enters these mice in their intestines as they groom themselves. These mice are otherwise germ-free, so they have no other commensals on them. And we can compare these monocolonized skin grafts with the germ-free mice that are raised in completely sterile conditions. What you can see is that we verified that this colonization protocol stably colonizes the skin of germ-free mice. So you can look before we start painting the mice, both the feces and the skin swabs remain clear when cultured for 24 hours. And then after painting these mice on the skin every other day for 10 days, and then stopping the painting process, the skin is stably colonized for at least 2 weeks in this germ-free condition. And so with that in mind, we then did the experiment where we took either a sterile skin graft or a Staph epi colonized one and transplanted them into germ-free hosts that lack their own microbiota. And what you can see very, very clearly is that indeed colonized skin grafts are rejected faster than sterile ones. And this was work done by Kevin Lay in the lab early on when they joined. And he showed that so indeed we know that the presence of a single commensal on this donor skin is sufficient to accelerate allograft rejection. And he went on to ask about one reason why this might be. And so what he did was, again, he took a male donor into a female recipient, so a minor mismatch allograft, same setup as before, except instead of watching for graft survival, he harvested the APCs from the skin and the draining lymph nodes and either looked at cytokines in these APCs by qPCR, or he took just the skin APCs, cultured them with a T cell subset called Maryland T cells that is directly responding to the male antigens on the allograft, and examined how those cells proliferated in response to the, or when co-cultured with the APCs. And what you can see first in these qPCR assays is that the skin draining lymph nodes really show no differences in the cytokines produced, but in the skin themselves, these skin APCs upregulate certain pro-inflammatory cytokines, and sure enough, are able to expand the Maryland T cells in co-culture in vitro much more than the APCs from the germ-free skin grafts. And so what this tells us is that indeed there's a role for microbial colonization in augmenting the effector phase of the anti-graft allo response. But what we really thought could also be going on is that potentially hosts could be mounting a graft damaging anti-commensal immune response actually in parallel to this anti-donor allo response. And so we asked that question and we said, well, could this be going on? And so starting there, we switched to a syngeneic system. And we did this in order to separate a potential anti-commensal immune response from a concurrent allo response. And so we took female grafts and put them into female hosts. And we set it up initially the same way. So we have a germ-free donor or a Staph epi colonized donor colonized in the exact same way. And then rather than going into germ-free hosts, we started using more kind of physiologically realistic mice, so specific pathogen-free mice, which do have their own commensal microbiota. And we gave these mice Bowie T cells, which are a CD8 transgenic T cell that responds to staph epi, and we labeled them with CFSE to watch their proliferation after transplantation. And what you can see is very clearly hosts do mount a T cell response to donor commensals, both in the lymph nodes, which you can see here, and in the skin graft. So this is taken 6 days after transplantation, and this is 10 days after transplantation. And so sure enough, in addition to, any allo response that could be going on, not in this system, but in general, in transplant situations, here you can see that indeed when the donor graft is colonized, you absolutely mount a T cell response to this donor commensals. Now the question is that really important or relevant? And so we next asked whether there was enough damage caused by these anti-commensal responses to be seen even in a syngeneic setting. And so what we did was we set up the experiment the same way, except rather than sacrificing the mice and looking at an exogenous set of cells that we've transplanted into the mice, we just monitored graft survival and relied on the host's native immune system to potentially drive any damage that we observe. And because these are syngeneic grafts, we couldn't use traditional rejection curves because we didn't anticipate that these grafts would be fully rejected. So instead, we decided to develop a scoring system where we monitored grafts for signs of chronic rejection or damage, where you get 1 point each for the presence of hair, pigment, a large graft size, and no red spots, for a maximum score of 4 points and a minimum score or rejected score of 0 points. And so what you can see is that when we do this experiment and we look in the green line at the staph epi colonized grafts and in the blue line at the germ-free grafts, there's this brief period of kind of minor transient damage that's very reproducible. And you can see that if you track these maximum damage scores in the mice that were colonized with staph epi, it's worse than the sterile grafts. But we thought about this a little bit more, and we were thinking about kind of what was most likely to be true in a transplantation setting. And what we know is that hosts, like recipients of transplants, have their own commensal microbiome. And in fact, even though everybody's microbiota composition is a little bit different, there's a lot of overlap between people, certainly between people who live in similar regions or countries as people who receive transplants in the United States do. A lot of these transplant runs are regional. So we thought that probably hosts would share commensals with the donor. And because staph epi isn't a native mouse commensal, we weren't really modeling that in the prior experiments. And so what we did was think about what those immune responses were. And we know from a number of papers that hosts, all people, harbor memory responses against their own commensals. And these memory responses can take a lot of different forms. They can be, usually there's kind of a symbiosis between the commensal and the self, but the cells themselves can have different phenotypes, especially in different contexts. And so in order to give hosts memory to this non-mouse commensal, we developed a system where we subcutaneously immunized mice with staph epi a month before transplantation to give the immune system time to kind of remember the bug. And we opted for the immunization approach just because it was a more consistent way of establishing memory than painting the mice, which would have been a lot more dependent on like construction in the mouse room or other shifts in the environment, and a lot more dependent on like very, very stable colonization. So this offered us a way of establishing memory. And we verify in the paper, this is not data I'll show you today, that this approach going subcutaneously does indeed create a very robust subset of anti-staph epi immune cells in both the skin and the draining lymph nodes, as well as a broad tissue resident memory T cell population in the skin. And so what we did was we took these hosts that had been immunized and we gave them either a staph epi colonized graft or a germ-free one, and scored them in the same way as in the other case. And what you can see very clearly, very reproducibly, is that in these syngeneic grafts, remember these are still devoid of any allo response, you see very significant damage in the hosts that have staph epi memory that receive a staph epi colonized graft. And this isn't just because of some infection due to memory or colonization, because these grafts don't get damaged anywhere close to as much. And it's clearly some impact of the combined memory and colonization that causes this damage. I will get back to this recovery in just a moment, but what you can see is that these grafts get damaged, and then around day 20, they start to recover back to baseline. And so this was macroscopically. We wanted to know whether this correlated with anything microscopically to give us a better sense of the pathology. And sure enough, what we can see, and this was analyzed by Dr. Hussein Sattar at the University of Chicago, who's a pathologist there, is that if you look on day 8, all the way through until day 25 post-transplantation at these grafts with in hosts with memory to the colonized skin graft, you see this dense infiltrate around the base of hair follicles, and sometimes in the more severe pathological conditions up the hair follicle, excuse me. And these cells are mostly a mix of lymphocytes, some monocytes, some neutrophils, all very consistently around these hair follicles, which is very interesting because that's where staph epi is known to reside, associated with the keratinocytes at the base of the follicles. And if you quantify per section the number of follicles that are inflamed, and you correlate it with the graft score, sure enough, the lower the macroscopic score, the more inflamed sites we see. This damage is very restricted to the graft. So if you look in the adjacent flank, you see no evidence of pathology. So we think that, what we're really seeing is this targeted immune response against staph epi that's driving some essentially collateral graft damage. And one key question, though, is why this phenotype goes away. And we had two hypotheses. The first was that because this isn't a stable commensal, once the graft that's colonized with staph epi gets transplanted into the mouse that has its own commensal microbiome, it might just get outcompeted over time. And the other hypothesis was that it might be getting actively killed in the hosts with memory. And so to test this, I just painted regular SPF mice that were either just wild type mice or had been given anti-staph epi memory with the bug for several days. And then I stopped painting and by PCR amplified for staph epi antigens over time. And what you can see in the green is that sure enough, over time, the bug goes away, becomes undetectable around day 32. But in the hosts with memory, it goes away much faster and is consistently lower amplification. And by day 17 or 18, which is interesting because this is where we see that the graphs start to improve, you see that the staph epi is mostly gone in the host with memory. So we think what's happening is that there is this target immune response that's killing the bug and by accident the graft. And remember, this is a syngeneic setting, so keep in the back of your head for later how this might play in an allogeneic setting. Okay. Importantly, staph epi is not the only bug where this happens. So if you use a commensal strain of Staph aureus and you do the same set of experiments, you can see that sure enough in the brown line, the hosts with memory that are colonized with Staph aureus behave the exact same way as with Staph epi. So we really think we are modeling and seeing something that's reproducible across many commensals and more broadly relevant than just a specificity of staph epi. So we next wanted to ask, why is this happening in this syngeneic system where we don't have an allo response? Like what cells are responsible for this commensal mediated damage? And we first turned to T cells. So what we did was to initially test this, we used TCR alpha knockout mice and we immunized them, or I should really say sensitized, because they don't have T cells, so their memory responses are expected to be slightly different, but they were sensitized in the same way with staph epi a month before transplantation with a colonized graft. And then we looked at survival using the same 4-point scale. In one set of experiments, we didn't do anything else. In another set of experiments, we added back naive T cells from a regular B6 mouse. These TCR alpha knockouts are on a B6 background. Naive cells that had never seen staph epi before. And in a third set of experiments, we added back in memory cells from SPF B6 mice that had been subcutaneously immunized with staph epi. And what you can see is if you compare in this orange line, this is our wild type mice line that I've shown you on previous slides. In the dark blue, you can see the hosts that lack all T cells that were sensitized with staph epi that receive a colonized graft do much better than when they have an intact T cell population. If you add back in naive T cells, similarly, they seem to be protected. And then if you add in memory cells, they start to do worse. And importantly, if you look in these mice that were sensitized, that are just TCR alpha knockout mice, no additional cells added, you can see by pathology that there's really histology, there's no infiltrates around hair follicles in the graphs that we looked at. And so they do seem to be protected, and whatever damage we see here seems to be not consistent with the damage that we see in these hosts with memory that get a colonized skin graft. Now, we really wanted to be a little bit more precise with this, because TCR alpha knockout mice, even when you're adding back in cells, they are immunologically just different because they lack, they're immunocompromised from birth. And so we wanted to really more precisely ask whether memory T cells against staph epi were necessary for this phenotype. And so what we did was we depleted CD4 and or CD8 cells using antibodies before immunization, and then throughout the immunization process. And then this allowed us to just deplete the cells at the time of memory induction. And then we allowed all of the CD4 and CD8 populations to recover. We monitored this by looking at CD4 and CD8 cells in the blood by flow before transplantation. And so these hosts now had intact immunity before memory induction, are now their CD4s and CD8s are recovered by this time. They just lack anti-staph epi memory, and we gave them a colonized skin graft. And what you can see on the left in the gray line is that in the hosts that were depleted of CD4s and CD8s in that way, they are almost completely protected from the damage you see when they have those populations intact at the time of immunization. It doesn't seem to be one or the other. Either CD4s or CD8s alone can mediate the damage, so they're probably both involved in some way. All right, so what subset of T cells, memory T cells, do we think is involved? So we reasoned that probably, given that we're inducing, or potentially, given that we're inducing kind of this memory responses, we know locally in the skin, there could be some role for tissue resident memory T cells. And so what we did is we set up kind of the host and the donor in the same way, except that the host, we gave the sphingosine 1-phosphate agonist, FTY720, a little bit before transplantation, and then continuing roughly weekly or every couple of days after transplantation, in order to essentially trap immune cells where they were. So we weren't getting any more naive cells or central memory cells coming from the lymph nodes, and any TRMs that were already in the skin presumably would stay there. And so when we did this experiment, what you can see is that these curves are entirely overlapping. So it does seem like the hosts that were treated with FTY720 are still able to damage the graft, suggesting that probably tissue resident cells, it could also be effector memory cells, are potentiating the damage, and putting this together with the previous slides most likely relates to tissue-resident memory T cells that are mediating this damage against these staph epi colonized grafts. Importantly, again, if you look microscopically, you see that in these mice that did receive FTY720, you see the pathology around hair follicles that looks the exact same as when they don't have FTY720, further suggesting that, again, this damage that we're seeing is the same in both, in both conditions. Okay, so what I've shown you so far is all in syngeneic system. But you're going to say, Isabella, people receive allografts and they get immunosuppressive drugs, and I agree with you. So what we did was we said, okay, let's just see what happens if we make both of those conditions true. So we did the same set of experiments where we gave the host memory and the donor staph epi, and our controls, of course. And we took either a male into a female graft, so a minor antigen mismatch, or a BALB/c into a B6 mouse, which is a major an MHC mismatch, so major mismatch. And then we gave these hosts either daily rapamycin, so an mTOR inhibitor, or daily tacrolimus, two different immunosuppressive drugs that hit different pathways, or different, yeah. So when we do these experiments, let's start with the rapamycin data. So if you look in the green line, this is your mouse that receives a staph epi colonized graft that doesn't have memory to staph epi, and this is a minor antigen mismatch, but that receives rapamycin. We know the rapamycin is working because if you look in the gray line, which is the same condition without rapamycin, the survival is prolonged. Now, if you compare the green and the orange lines, what you can see is that very clearly the presence of anti-staph epi memory, even with immunosuppression in the case of an allograft, is sufficient to accelerate allograft loss. And so that's the first conclusion. First of all, hosts with anti-commensal memory have worse prognosis for, or reject their grafts more quickly than hosts that lack this memory, even given immunosuppression. And second important point is if you compare the orange and the purple lines, you can see that essentially in the hosts with anti-commensal memory, the rapamycin has no effect. These lines are completely overlapping, suggesting that whatever is driving this accelerated graft loss is resisting immunosuppression. And the same thing was true with tacrolimus in both the minor and the major mismatch settings. So here, what you can see if you compare the blue line, which is hosts that just have memory that got a germ-free graft, with the orange line, which is hosts with memory that got a colonized graft, and both got tacrolimus, the additional presence of colonization on the graft in hosts with memory is sufficient to accelerate rejection, and hosts in the minor and the major mismatch case show no difference if they have memory, whether or not they receive tacrolimus. And actually in this situation, the host that did not, that received immunosuppression did significantly worse. And so summing this all up, what we can see, what we propose as a model is that these organs that do so much worse in the clinic are colonized with their own commensal microbiome, which means that they're bringing over, so in addition to kind of what the lab has shown before, which is that there is probably some role of kind of augmenting the allo response. There's also very clearly, an anti-commensal immune response that's playing some role. So it comes over with donor mammalian cells and donor commensals on this graft, which is two, types of antigens. The hosts are most likely to harbor some anti-commensal memory. And so in addition to this host versus graft response, which is what we conventionally hit with immunosuppression and which is successfully dampened that way, largely because usually it's a naive response or so we think. This host versus commensal immunity, which is also being mounted, is sufficient to accelerate allograft loss and resist immunosuppression, presumably because it's driven by tissue resident memory cells, or at least memory cells. And so when we think about this, it's a bit of a puzzling question, kind of where to go from here, right? I think the most clear potential solution is that there might be some role for sterilizing organs before transplantation, maybe giving, and this is some work that Marissa is starting to do with a lung transplant surgeon at UChicago, is thinking about practically how you might be able to perfuse organs or intratracheally deliver antibiotics in that reperfusion phase, and how to do this in a way that you don't set yourself up for opportunistic infections post-transplantation when these hosts are immunosuppressed. And another idea would be to think about what happens if you establish tolerance to donor commensals in the neonatal period. So we know from work from like Tiffany Scharf, Yasmin Belkade, and others that if you if you expose mice, the stuff would be a little bit later in life, and this happens also in people, like we go to new environments, we get exposed to new commensals. Often those responses aren't quite as tolerogenic as they are if you are exposed to these things younger in life. Even though it's still a commensal, they are a little bit more poised to be pro-inflammatory should like a barrier insult happen, like a cut or something, or surgery. And so here we would give stuff FB to neonatal mice, start painting them like from birth and see if they are in any way protected. And then I think with all of this in mind, there's also room to think about mechanism and why is it that these memory responses are resisting immunosuppression. And to understand that, I think understanding things like how these cells might be moving into new tissue, exactly what the reactivation requirements are, et cetera, are kind of things to be thinking about. And so with that, I just have to thank the Alegre lab. I started in the lab as an undergraduate, and it takes a village to raise a young scientist. So every single person in this picture played a huge role in this project in some way. Most especially, so Kevin trained me in the lab. He's since graduated. Caroline and Christine did a lot of work on the microbiota before I even joined the lab. Martine did all of the rebuttal experiments. So I'd, left the lab by that point, and he really, a huge part of why this paper has kind of insight into immunosuppression and so on is because of experiments that he did. Ying and Luchu are our microsurgeons. The lab would not run without them. And Marissa, of course, is our fearless leader and a wonderful mentor. These are all of our collaborators, both at UChicago, the NIH, UCSF, our funding sources. And then a huge thank you to the 2 notobiotic technicians at UChicago who really, this would not be possible without. And so with that, I would like to take any questions. Thanks.
Thanks, Isabella. That was a great presentation. I have some questions, but I'll first go to the Q&A. Jay Fishman says, very nice study. Do you anticipate that the pathogen matters, for instance, commensal versus pathogen? If not, all infections might augment graft loss. We know this from clinical experience, although not always recognized as rejection. Conversely, GI microbes might generate Tregs as well as T effector cells and reduce inflammation. Any comments?
Yeah, so I think first of all, absolutely right. And actually another project in the Alegre lab looks at the roles of actually infectious microbes in breaking tolerance and breaking, whatever tenuous set of kind of immune suppression we can create with drugs or with other kind of tolerogenic protocols that we could use in a lab more so than clinically. And we know that infectious pathogens can absolutely break that. And that, that's there. I'm thinking about more of the allo response, right? So that's that piece. I think certainly, secondly, a pure infection, we know can just make an organ fail for whatever reason, or be so deleterious to somebody's health that for reasons that have nothing to do, with or not nothing, but less to do with an allo response. They just will die, and that would simply also get read out as graft, failure in some depending what metrics people are using. So that's to the pathogen point. I think my anticipation is that this happens with a lot of different commensals, this phenomenon of kind of having memory and then matching. I'm sure some more than others, but I also think that it's more so the phenomenon and the general principles that are important, because you're never, I don't think, going to be able to map like individual commensals and either match or mismatch them. I think that practically that's not possible. So I think where I think about this is thinking about how you, like general mechanism. And I suspect that this is happening for a lot of commensals.
Great. So I have a question about the tissue resident memory part of this study. So it seems like, I wonder if you ever addressed, it looks like you were largely modulating memory in the host. Did you do any studies, or were there any aspects of the studies that looked at modulating memory in the donor? The idea of, and if I understand the model correctly, that you're inducing memory in the host, but can you induce memory in the donor that then takes up residence in the skin, which then gets grafted, if that makes sense? And then versus in the host, presumably those resident memory cells in the skin are moving into the graft without having to go into circulation and home back in that way. Am I thinking about that correctly?
Yeah, I mean, I think so to take the second part of the question, like how are the cells getting there? In a skin graft, you can absolutely imagine like lateral migration. I think for something like a lung or something where you're just doing one lobe and there's not as much directly adjacent tissue, our experiments with FTY 720 in theory would kind of prevent or significantly reduce that recirculation. But I actually think, like, if you look at work from like out of the Mazepus lab and others, it's pretty clear that tissue resident memory cells recirculate in response to exposure. And I suspect that that could also be going on just practically. And so I envision probably both. I think, now, can you remind me? So that's sort of the second part of your question. Can you remind me the first part?
I mean, to what extent do you think in your model or in general, memory T cells coming, passenger, cells, memory T cells coming from the graft, from the donor, could also mediate this problem?
Yeah, so the short answer is I don't know. We really didn't look at that at all. Almost like a graft versus host against commensals.
Right.
It's a bit more of it's a bit less obvious to me, the mechanism there, unless the hypothesis is that essentially you bring in donor cells that then attack the host microbes that start to colonize the graft, and because they're still matched, it's kind of actually sort of the same mechanism, in the sense that maybe it's just like lateral attacking. That's an interesting question that we just didn't look at, but I think could be.
Another question from the chat is asking, do you know anything more detailed about the T cells that are responding against the commensal? Are they Th1 cells, Th17 cells, et cetera? Any other details about that response?
No, we didn't look. I at one point did a set of experiments that I didn't get to work very well, looking at kind of what cytokines they were making, or so what their effector programs were, and the data was just not. It was probably my error more than anything else, to be completely honest with you. But it just wasn't clear to me, and there certainly wasn't one that was dominant over the others. But it's something that's worth looking into. I will say, I think we know that for things like lung transplants, the allo response tends to be more skewed, I think, to like type 17. So there are differences in an allo, what happens, and I'd be curious to know, kind of to look back at how some of those studies were done and see what proportion of those cells are like true allo versus like have a commensal reactivity. Yeah,
it gets into an interesting question of that, what might that interplay be between the anti-commensal and the allo T cells. Seems like in your model, the memory against the commensal is probably setting up a situation that facilitates homing of alloreactive, presumably naive and in many hosts T cells into the graft and priming. So another question from the Q&A: do you expect this type of reaction against commensal microbes in skin and mucosa to play in transplant of distal organs like kidney or heart?
Yeah. So I think that gets into an interesting question. So for example, we know that if you modulate the gut microbiome, you can have an impact on skin graft outcomes, et cetera, right, et cetera. Like, I think it's very clear that gut immunity and has these effects all over the body. So I think broadly, yes, but I don't know, thinking about a model where if you're really thinking about the commensal specific cells, right? In my mind, and this is because of data I showed and data I didn't show, I'm pretty convinced that they're antigen specific, or at least relatively antigen specific. Like one strain of Staph epi or another, slightly different, probably they could cross react, but like Staph epi to E. coli or something, they're not reacting. And if you take that as your model where you're saying it's antigen specific, it just to me would depend on, like, is the heart, because you're cutting open a skin wound and presumably exposing it to microbes, is it when it's getting transplanted bringing in some, like, maybe, but I think I don't think of our model as being quite as systemic as like a gut effect. But maybe it looks like Marissa.
Yeah, maybe I can chime in, because otherwise I feel like I'm doing nothing here. Isabella has all the answers. But I think two answers. I think the intestinal microbiota is helping antigen-presenting cells be poised in a way that can present alloantigens better. So in that sense, all the gut microbiota is going to help autoimmune responses also to sterile organs, but in a more generic way. The only way I could see the answer to the question as to whether a specific anti-commensal T cell would be able to damage a distal organ that's not colonized, like a kidney or a heart, would be cross-reactivity. And I'm sure that can happen, and people are starting to look at the peptidome of all the microbes in the body. And there are many motifs that would fit into host MHC class 1 or class 2. So I'm sure there are going to be instances where an antimicrobial response would be cross-reactive to some kind of protein expressed in an organ. But it would be cross-reactivity. I don't think it would be, or the other way you could think about it, and Isabella worked a lot about that, is bystander responses, that if you have an immune response to a commensal and you have a lot of, say, IL-6 production or other cytokines that could enhance the proliferation of an ongoing alloresponse, that would be another way. So you have the poisoning of APCs, inflammatory cytokines that can enhance proliferation of allo immunity, and then you have cross-reactivity. Those would be the three mechanisms I could think of.
And then for both of you, just, for my curiosity and not really my area, is the memory T cell pool, is it resistant to most of the immunosuppressive regimens that are used in the clinic? Are they generally already poised to function and can still do their job in the face of, you showed rapamycin as an example of this. Any comments on just why memory T cells might, I know this is a large field of study in transplant, but might be more difficult to deal with from an immunosuppression point of view?
Yeah, I mean, so my kind of basic level of understanding of requirements in general for memory cells to get reactivated, ignoring immunosuppression for a second, is that typically they have a lower threshold for activation. And so, some of the kind of differentiated, very poised cells already exist, right? So you don't need to, whereas, so I think that's sort of what I think. And then many of the immunosuppressive agents hit like the cell proliferation cascades that are much more active out of freshly dividing cells. And I think there, and I suspect also stuff like how much co-stimulation they need to be activated. But Marissa has, I'm sure, a much more precise answer to that question. But
no, I think that's right. I think memory cells are just more difficult to immunosuppress, and memory alloreactive T cells, we think are major antagonists to the induction of transplantation tolerance. They're just more difficult to immunosuppress with conventional immunosuppression, and also with the agents that we use to try and induce tolerance. So I suspect if the mice had memory to alloantigens in addition to memory to the commensals, they would be even more resistant to immunosuppression. I think a key question for the field is how can we suppress memory cells, whether they're anti-commensal or allo reactive.
Right.
How can we reprogram them? I think there's a couple of there's another question that I see from Pam Jacobson in the chat. Does rapamycin have activity against staph? Do you think there would be value in trying to match immunosuppression to the predominant commensal?
You mean acting like an antibiotic? Is that the question?
Yeah, or anti-replication. Like rapamycin can have activity against viral replication, and maybe immunosuppressive drug of choice. If you are trying to immunosuppress and there's a viral infection, you can kill two birds with one stone. We have not tried to see if there was less replication. I would suspect not, because if it would inhibit replication of staph, then we would have seen a benefit. Probably in the colonized skin grafts, they would have behaved more like the germ-free skin grafts, or maybe in between, whereas they seem to be completely resistant. So I suspect it doesn't affect replication of staph, but we haven't tested that.
Okay, if there's no further questions in either the Q&A panel or the chat, I just again thank Isabella for taking the time out of her busy schedule and presenting.
Oh, and I should emphasize that Isabella did all of this while she was an undergraduate student in my lab. So she worked for 4 years in my lab and, spearheaded this project completely, and then stayed an extra year as a tech to where she was applying to MD PhD programs. So, I think it's quite remarkable that she can first author paper for a study that I think is really, really cool.
Yeah, great work. Okay, thanks everyone for attending. Thanks again to Marissa and Isabella. And stay tuned for our next COTS AST sponsored journal club in the future.
Thank you so much. AST would like to thank Ms. Pirozzolo, Dr. Alegre, and Dr. Wells, and all of our attendees for today's session. Please remember to complete the evaluation survey that shows up in Zoom after the webinar is complete, and visit myast.org/journal club to view our video archives and register for upcoming journal clubs. To learn more about the AST's community of transplant scientists, please visit myast.org/COPS or connect directly to the individual COP hubs. Thank you again for today's session.
Thanks, everybody.